Cytokines are a broad and loose category of small proteins, around 20 kilodaltons, that are important in cell signaling. Cytokines are peptides and cannot cross the lipid bilayer of cells to enter the cytoplasm. Cytokines have been shown to be involved in autocrine, paracrine, and endocrine signaling as immunomodulating agents. Their definite distinction from hormones is still part of an ongoing research. Cytokines include chemokines, interferons, interleukins, lymphokines, and tumor necrosis factors, but generally not hormones or growth factors, despite some overlap in terminology. Cytokines are produced by a broad range of cells. These include immune cells like macrophages, B lymphocytes, T lymphocytes, and mast cells, as well as endothelial cells, fibroblasts, and various stromal cells. A given cytokine may be produced by more than one type of cell. They act through receptors and are especially important in the immune system. Cytokines modulate the balance between humoral and cell based immune responses, and they regulate the maturation, growth, and responsiveness of the particular cell populations. Some cytokines enhance or inhibit the action of other cytokines in complex ways. They are different from hormones, which are also important in cell signaling molecules, in that hormones circulate in higher concentrations and tend to be made by specific kind of cells. They are important in health and disease, specifically in host responses to infection, immune response, inflammation, trauma, sepsis, cancer, and reproduction. The word comes from Greek, cyto from Greek, kaitos means cavity, and kines from Greek, kainos means movement. Discovery of cytokines. Interferon alpha, an interferon type 1, was identified in 1957 as a protein that interfered with viral replication. The activity of interferon gamma, the sole member of the interferon type 2 class, was described in 1965. This was the first identified lymphocyte derived mediator. Macrophage migration inhibitory factor was identified simultaneously in 1966 by John David and Barry Bloom. In 1969, Dudley Dumont proposed the term lymphokine to describe proteins secreted from lymphocytes and later protein derived from macrophages and monocytes. In culture, they were called monokines. In 74, Stanley Cohen published an article describing the production of MIF in virus infected allantoic membrane and kidney cells, showing its production is not limited to immune cells. This led to his proposal of the term cytokine. Okawa described the early acting growth factors, intermediate acting growth factors, and late acting growth factors. Each cytokine has a matching cell surface receptor. Subsequent cascades of intracellular signaling then alter the cell function. This may include the upregulation or downregulation of several genes and their transcription factors, resulting in the production of other cytokines, an increase in the number of surface receptors for other molecules, or the suppression of their own effect by feedback inhibition. The effect of a particular cytokine on a given cell depends on the cytokine, its extracellular abundance, the presence and abundance of the complementary receptor on the cell surface, and downstream signals activated by receptor binding. These last two factors can vary by cell type. Cytokines are characterized by considerable redundancy in many cytokines appear to share similar functions. It seems to be a paradox that cytokines binding to antibodies have a stronger immune effect than the cytokines alone. This may lead to lower therapeutic doses. SAID showed that inflammatory cytokines cause an interleukin 10 dependent inhibition of T cell expansion and function by upregulating PD-1 levels on monocytes, which lead to interleukin 10 production by monocytes after binding of PD-1 by PDL. Adverse reactions to cytokines are characterized by local inflammation and or ulceration at the injection sites. Occasionally, reactions are seen with more widespread papular eruption. Now, this is a comprehensive review of cytokines.